Hey guys, I want to talk to you about a result that was published this past month in the journal PNAS. And the paper is changing the paradigm of transcription, which is the conversion of DNA that encodes genetic information to RNA, which is an intermediate between that genetic information and protein, and has some function to it. It's changing the way that people are going to think about transcription, because after transcription is terminated, the RNA polymerase doesn't come off the DNA. Previous studies of RNA polymerase using single molecule microscopy show that RNA polymerase can remain bound to DNA hundreds of seconds after transcription. And this group, led by Jeff Gels and Jane Kondev from Brandeis University, showed that RNA can initiate transcriptions thousands of base pairs after termination. So the RNA polymerase terminates transcriptions and releases the RNA, but remains bound on that DNA strand and diffuses or slides down the DNA to the initiation site. And this isn't the only way that genes are correlated during transcription, but it is one way. Transcription measurements show correlation between the distance of the genes and their expression, as well as the initiation factor concentrations, which are required for the initiation of transcription. There are other ways that gene transcription can be correlated, and this can happen through common regulatory proteins that initiate transcription, like the initiation factor sigma 70 in E. coli. But gene expression can also be correlated due to DNA supercoiling. This can actually cause the expression of genes that are far away from that gene that is currently being expressed, but the supercoiling is actually caused by that transcription. It's called transcription-coupled DNA supercoiling, and I just learned about it in this paper. Another way to get correlation of gene expression is by terminator read-through, and this is when an RNA polymerase fails to read the termination sequence and continues to transcribe RNA through the DNA between the two genes, which are introns, and it goes all the way to the start of the next operon. An operon is either a single or multiple genes that are transcribed together. They're transcribed together, but they may not be translated together. It could be separate genes that become one long mRNA, and it may or may not have multiple protein products. So this group modeled the process. They knew the RNA diffuses along the DNA with the coefficient D. It can dissociate or can bind an initiation factor sigma 70, and it can dissociate subsequently, or it can bind sigma 70 and initiate transcription at the initiation site, the promoter. Then they experimentally determined the kinetic parameters for each of these. They knew the diffusion coefficient. They determined off rate, the rate of release of the RNA polymerase from the DNA. And then measured the binding of the sigma 70, and then they measured the off rate of the polymerase with sigma 70. They then plugged the values into the model, and they predicted the distances and times over which RNA polymerase could initiate. Previous research had shown that RNA polymerase stalled for certain molecules, and those researchers had attributed this to binding to DNA ends. So the researchers in this paper used DNA without promoters. They used Cy5 labeled DNA and also a dye labeled RNA polymerase enzyme with a green dye, and they alternated imaging of the two so they could locate the circular DNA and the RNA polymerase. And they restricted analysis to DNA with only a single RNA polymerase bound. And they simply measured the dwell time of the RNA polymerase on DNA. So they had a signal present where the polymerase was right next to the signal for the DNA, and this DNA is immobilized on a glass surface, by the way. By measuring this dwell time at different concentrations of sigma, they were actually able to find the probability density of an RNA polymerase finding a subsequent operon, and also the coupling efficiency at different concentrations of sigma. The coupling efficiency is the coupling of the expression of two genes that are nearby each other. using the same RNA polymerase for both. And they found with their experiments and their calculations that there is a high probability that an RNA polymerase will extend past termination, bind sigma, and reinitiate transcription at the next operon. We're talking like 10 to 80% coupling efficiency. This means 10 to 80% of the time, as long as it's on the order of 1,000 or several thousand base pairs or less, you're gonna have that kind of probability of coupling between two subsequent genes. So what, you may ask? Why is this important? I think it significantly changes the way we think about transcription, and it's hard to say what actual impact it will have on the world. It may cause an innovative person to think of a way to use this in a way that may improve medicine or improve our techniques to do biological research. It may just lead to the next discovery as science is building upon itself constantly. Thanks so much for watching. Have a great week. Take care.